All right, let's dive right into this explainer on the biology, history, and terrifying cellular heist executed by the infamous anthrax bacterium. We hear the word anthrax thrown around a lot, usually in the context of modern thrillers or biowarfare. But the true story of this pathogen is an absolute masterclass in microscopic survival and sabotage. And as we'll see, it fundamentally shaped modern medicine as we know it. We're going to use a zoom-in, zoom-out approach today. Our agenda starts with the macro history. Part 1, the harmless and the deadly, followed by Part 2, the dawn of microbiology. Then we'll zoom way into the cellular level for Part 3, a microscopic Trojan horse, before pulling back out for Part 4, 3 paths of infection. Finally, we'll wrap up with Part 5, beating the scourge. Okay, Part 1, the harmless and the deadly, a tale of two bacilli. To really understand our culprit, we first need to look at its surprisingly mundane family tree. So the word bacillus actually comes from the Latin baculum, meaning staff or walking stick, which makes sense given the rod-like shape. There are over 250 species of bacillus, and for the most part, they are just ubiquitous, everyday soil dwellers. Seriously, if you go out and scoop up a handful of dirt from your backyard right now, you are almost guaranteed to find them just quietly going about their business. But here's where it gets kind of wild. There is a massive, stark contrast within this family. On one side, we have mostly harmless strains. You might even have some in your garage, like Bacillus thuringiensis, which is a super common organic bug spray, or Bacillus cereus, which is that bug that might give you a mild case of food poisoning if you eat leftover rice that's been sitting out. But then, on the flip side, we have Bacillus anthracis. Eons ago, it actually evolved from that mild food poisoning bug, picked up some incredibly deadly genetic traits, and transformed into a highly lethal, aggressive pathogen that jumps from animals to humans. A true zoonotic killer. Moving on to Part 2, the dawn of microbiology, proving germ theory. Let's see how this exact disease essentially birthed modern infectious disease science. Back in the 1870s, scientists were literally arguing over whether bacteria actually caused disease or if they were just the rotting aftermath of it. Well, Robert Koch and Louis Pasteur used anthrax to definitively prove the germ theory. In 1876, Koch was looking into these massive farm animal die-offs. Because artificial petri dishes weren't even a thing yet, he resourcefully used the vitreous humor, literally the jelly inside a cow's eyeball, to successfully cultivate pure anthrax bacteria. Then, a few years later, in 1881, Louis Pasteur dramatically proved his new vaccine worked by injecting 50 sheep with live anthrax. The vaccinated sheep lived, and the unvaccinated ones perished. So the big takeaway here is that by continually infecting lab animals with lab-grown anthrax, Koch established these foundational postulates that scientists still use today. He proved beyond a shadow of a doubt that a specific microbe causes a specific disease. This single organism essentially kicked off the golden age of microbiology. All right, Part 3, a microscopic Trojan horse, cellular sabotage. We are now zooming all the way down to observe the highly sophisticated, weaponized heist that makes anthrax so deadly at a cellular level. Remember how I said earlier that Bacillus anthracis is almost identical to its harmless cousins? The entire difference basically comes down to plasmids. These are independent, usually circular DNA strands that sit apart from the main bacterial chromosome, and they code for very specific proteins. Think of them like, well, deadly expansion packs for the bacteria. Our pathogen possesses 2 of these plasmids, and they carry the exact blueprints for its microscopic weapons. Let's break down this arsenal. Plasmid POX2 codes for a protective capsule. This acts like a cloaking device, hiding the bacteria from our immune system's phagocytes. Meanwhile, Plasmid POX1 codes for 3 specific things: protective antigen, lethal factor, and edema factor. This combined arsenal allows a bacteria to completely blindside our first responders, specialized white blood cells like neutrophils, macrophages, and dendritic cells. It literally stops our innate immune system dead in its tracks. And this is exactly how that Trojan horse maneuver I mentioned plays out. First, the bacterium tricks the host cell into binding with the protective antigen, which then forms a literal molecular barge. Next, the lethal and edema factors hop on board, and the host cell mistakenly swallows the whole entire thing into an endosome. But once it's inside and the environment gets acidic, the barge activates. It bores a hole straight through the membrane and unleashes the deadly toxins from the inside. The cell has essentially pulled in its own assassins. Once those toxins are loose in the cytoplasm, the absolute sabotage begins. Lethal toxin blocks MEK enzymes, which completely disrupts the cell's MAPK signals. Think of these signals as the cell's emergency communication lines to the nucleus. By cutting those phone lines, lethal toxin paralyzes the cell, preventing it from calling for backup and giving the bacteria totally free rein to multiply furiously. At the exact same time, edema toxin is attacking a completely different set of signals, protein kinase A. Imagine breaking a vital cellular pressure valve. This causes a massive buildup of a molecule called cyclic AMP, shredding normal cell function and resulting in massive fluid loss and swelling, or edema. Together, these two toxins execute a perfectly coordinated 1-2 punch, destroying cellular function and flooding the surrounding tissues. Okay, part 4. 3 paths of infection, routes into the body. Let's zoom back out to the human scale and see how this microscopic threat actually gets inside us. Anthrax survives in the environment by forming spores. Interestingly, Bacillus anthracis doesn't actually form spores inside a living host. But the second an infected animal dies and the bacteria are exposed to open air, the organism locks itself down into this practically invincible, dormant bunker state. 50 years! That is how long these incredibly hardy spores can remain viable in the soil, just waiting for another animal to graze over them. This extreme resilience is exactly what led ancient shepherds to wisely avoid grazing their flocks in specific pastures that they naturally came to call cursed fields. When a human encounters these spores, there are 3 primary routes of infection. A cutaneous infection creates a painless black scar, which, fun fact, is actually where we get the name anthrax, from the Greek word for coal. A GI infection from eating undercooked infected meat causes intense abdominal pain. But it's the inhalation route, breathing the spores directly into the lungs, that is by far the most aggressive and vicious. Without treatment, the mortality rate for inhalation anthrax approaches 100%. Let's see how this builds into the ultimate biological trap. When spores enter the lungs, our macrophage immune cells pick them up, but they don't destroy them. The sheer diabolical genius here is that the anthrax's edema toxin actually counteracts the lethal toxin, actively keeping the host cell alive just long enough to act as a free Uber ride straight to the lymph node. Once it reaches that sweet spot, the bacteria break out, multiply wildly, and dump massive amounts of toxin directly into the bloodstream. This specific inhalation trap caused absolute devastation during the Industrial Revolution. In 1837, in Bradford, England, mill workers began mysteriously dying in mass. It turns out they were inhaling airborne spores from contaminated, imported mohair and alpaca hides. It became known as wool sorter's disease, and it actually took decades and the discoveries of Robert Koch for the world to finally realize these industrial workers were falling victim to inhalation anthrax. Which brings us to part 5, beating the scourge, modern defenses. After all that cellular destruction and dark history, let's explore how modern medicine finally learned to fight back. Our modern defense basically requires a 2-pronged approach. First, antibiotics like ciprofloxacin killed the bacteria by stopping their protein synthesis. However, because the toxins just keep right on destroying things even after the bacteria are dead, combining antibiotics with antitoxins is absolutely vital for advanced cases. These antitoxins are laboratory-made antibodies that literally plug up that molecular ferry, the protective antigen, completely blocking the toxins from ever getting inside our cells. And thanks to Max Stern's 1934 vaccine for animals, anthrax is now incredibly rare in developed countries. We've mostly banished the natural threat from our farms and factories, but the organism's terrifying efficiency and near 100% inhalation mortality rate means we really must always remain vigilant against its potential for weaponization. I really hope this explainer gave you a deep appreciation for the complex, invisible wars happening every single day at the cellular level. I mean, if a simple soil bacterium can evolve into such a flawlessly executed microscopic heist, literally turning our own immune system into a taxi service for its lethal payload, it absolutely leaves you wondering, what other dormant threats are quietly waiting in the cursed fields of our world? Thanks for joining me on this deep dive. Keep questioning, and I'll catch you on the next explainer.